Okay, so maybe we can go to the... Okay, so yesterday we talked a little bit about population genetic theories of aging. And so I introduced you to this idea by Peter Medawar, the idea being that there's alleles or mutations that are effectively neutral at young age, so they don't have any effects on fitness when we're young, but they might have detrimental effects when we're old. And in an environment where most organisms are, let's say, already dead, by the time they could experience those effects, these mutations can just accumulate in a genome and they will not be weeded out by selection. So this is like one idea. And in fact, we have empirical evidence that such alleles or mutations really exist. Medawar also had a similar idea in his book that was later taken up by this guy here, George Williams, which is called antagonistic pleiotropy. The name is a little bit abstract, it's not so important to remember it, but the idea is very similar. Let's imagine a mutation that occurs at random in our genome, and it's going to have bad effects when we're chronologically old. But let's imagine that this very same mutation has good effects when we're young. And so selection would favor this mutation because it has good effects when we're quite young. It might have good effects on how well we can survive or reproduce and so forth. But at the same time, it's going to have a cost when we're old. And the logic is very much the same. If this mutation is, the negative effects of this mutation are expressed sufficiently late in life, then these late life effects are somehow inconsequential. They don't really matter. But the mutation is being favored because it's good early in life. And in fact, we do have many examples of such mutations, including in humans. Just to maybe make one example, this is not directly about aging, but it has to do with aging indirectly, which is, for example, breast cancer risk in women. There's like a gene that is called BRCA1, which if you have a sort of deleterious mutation in this gene, it increases the risk for developing breast cancer in women. And there's particular mutations in this gene that segregate in populations of humans where if you have this allele, unfortunately increases the risk that you will have breast cancer, but at the same time, when women are young, it tends to increase their fertility. So this is sort of an antagonistic pleiotropic effect. It's a beneficial effect early on in life, coupled with a bad effect late in life. And again, in a world that is very dangerous, where mortality from the environment by predation and pathogens and things like that is high, such a mutation, the bad effects would not be really consequential. They would not be seen by selection, but this mutation would be favored because it has early effects that are positive. So here under this model, aging evolves through the accumulation of alleles that are beneficial early in life, but deleterious late in life. And so this is also actually a genetical theory of trade-offs, because like many fitness components or life history traits that are relevant for our fitness, things like development, fertility, how long we live, and all these things, they often trade off with each other. So selection tries to sort of maximize fitness, but it's a constrained optimization problem, because maybe you can gain fitness by being more fertile, right? So that should like increase your fitness, but at the same time, the more fertile you are, maybe the less long-lived you will be, because there's a trade-off often between reproduction and how long you live. This is even the case in humans. In humans, we would think, I mean, we have modern hygiene and technology and nutrition. Selection does not act on us, blah, blah, blah. But this is not really the case. Even in humans, where we can measure this, we see that people who have more children tend to live less long. And that this relationship is not even, it's not only environmental, because like, having children takes up energy and time, it makes us tired and so forth. As I know from own experience, it's actually a genetical relationship. So we can even establish that this is a genetical relationship, that like in women that have more children, they tend to live shorter. And the same is true, remarkably, for men. And making eggs and bearing a child is energetically very costly. But sperm is super cheap, right? And so it's stunning that even in men, there's a negative genetic correlation, we say, between reproduction and lifespan. This has been established in a study in Framingham, Massachusetts. This study was started to sort of study cardiovascular health of people, and it's a longitudinal study that has been done for several decades. And they collected all these health parameters of people and DNA samples and all these other things. And so they know how long people lived, how many children they had, and all these things. And so evolutionary biologists actually worked with these data sets, and they found these sort of negative correlations between reproduction and longevity.
Can you explain the curve of strength of selection again?
Mm-hmm.
Because I understand the general trend that as you age, you
right.
become perhaps less motile. You already had offspring, so the
yes.
so the selection will
yes.
have acted also already. But there is a plateau in the beginning, and I do not really
yes.
have a clear explanation
yes.
of why it is
right.
a plateau.
Right. Okay. So these two curves here are idealized. And the upper one, the solid line, is sort of imaginary force of selection acting on one component of fitness, which is survival. And for fecundity and fertility, it actually looks a little bit different. So it depends on the fitness component. But in general, so this was like worked out by, maybe I'm going to go to the blackboard, by Hamilton in 1966 in this paper in Journal of Theoretical Biology. And he sort of calculated these things. So Ronald Fisher in 1930 had this book, The Genetical Theory of Natural Selection. In that book, Fisher had already this notion that there might be something that is similar to the force or strength of selection. There's a concept that is called reproductive value, which is a measure that we can calculate based on an equation that's called the Euler-Lotka equation. I don't want to go into the details, but so reproductive value intuitively is a measure of how many offspring we can expect to have as a function of how old we are. And so we can calculate this. So for example, we can calculate things like life expectancy at birth. So for people born in 2022, we can follow them through time, and we can then sort of calculate what is when they're born in 2022, how long can they expect to live, in the future? And reproductive value is something similar. This measures how many offspring you have and how many offspring you're likely to gonna have in the future. Now, this reproductive value Fisher calculated, he derived theoretically, and then he calculated using data from women in Australia. He calculated their reproductive value as a function of their age, using data from women from 1901, I think. And so what he realized is like, okay, so if you plot that curve, it's like this function, and you plot it as a function of age, then reproductive value tends to go up as you approach sexual maturation in women. So the expectation of having a certain number of offspring is going up as you approach sexual maturity. And then it sort of has a maximum around when you're sexually mature. And then he realized that it's coming down again as you get older. It's quite intuitive. We know that the probability that a 70-year-old woman will have children is okay. I'm not sure it's zero, but it might be very close to zero, effectively zero. So it's intuitive that it comes down. And he thought that maybe, because selection really cares about how many offspring we have, that this is sort of proportional, this curve, to the force of selection. And Hamilton formalized this force of selection thing and essentially calculated something that we could call sensitivity or elasticity, but it's like a sensitivity measure. It's like a partial derivative where you ask how much does fitness, let's say omega, change if you change, for example, survival probability, right, as as a function of age. And so Hamilton calculated these things and sometimes also used data from humans to sort of like look at these measures. And so you could like do something similar for fecundity or fertility, but often in aging we look at survival or mortality. And so he could like look at this partial derivative, which is sort of proportional to the strength of selection. And he saw like, okay, so this will, you know, this will somehow come down, you know, as we age. And he noticed, or people noticed that this comes down, starts to decline after age at first reproduction when we talk about mortality, but is rather constant early on in life. But of course the details depend on the organism, and so in principle for many organisms we have these data to be able to calculate this, but it's actually interesting that I haven't seen any systematic analysis where people have demographic data to systematically quantify this for different animals. But so the idea is really that the force of selection declines, but as I've mentioned yesterday, we know that there's organisms that as far as we can tell don't age, or they age very slowly. And so for them the curve must look quite different, right? For them the curve must be such that the force of selection stays constant much longer, right? Before it comes down maybe at some point. Or maybe it doesn't even come down. Yeah. So these are two genetical theories about how aging might evolve. And when these people dreamt up these theories, like Medawar and Williams, we only had the theory. We didn't have any proof for the existence of such alleles. But nowadays we can do experiments to really show that such mutations exist. And for a long time, molecular and developmental biologists thought like it's going to be impossible to study aging. Aging is too complex. It's just like we're a complex machine and there's so many things that can go wrong with this machine. Why would there even be specific genetic factors that like determine the rate of aging? And so people were really skeptical about being able to find genes or mutations that affect how long organisms live. They didn't really believe that this would be possible. But on the other hand, people had in a way already done something similar, because like some geneticists said, we want to study the genetic basis of development. Rather than doing embryology, we're going to use genetics to see whether we can understand something about development. And development is sort of like, well, almost the antithesis of aging. It's a constructive process where from a single oocyte that's fertilized, you're building a complex multicellular organism. And it works, I mean, as we know from experience, it works perfectly well in most cases. And so it's such an incredibly tightly regulated process, and people manage to use genetics to sort of uncover how it works. And so that led to a Nobel Prize for a couple of people, including Christiane Nüsslein Vollhardt. And they had done a mutant screen in Drosophila. They looked at a simple developmental phenotype, but they were the first to do a systematic mutational screen of developmental defects, and they managed to find these genes, many of which we have as well in our bodies. And they really showed as a proof of principle that you can use genetics to understand development. And so that led some developmental biologists to say, maybe we can do the same thing for aging. But at the beginning, people were very skeptical. But so then some people tried this, and it actually worked. And so like some of the first papers appeared in the early 80s. So this guy here, who is not that famous, but he was like the first to actually sort of do this. He used a little nematode worm, C. elegans. It's like this little worm that's like 2.5 millimeters long or something. And so he just created random mutant worms using mutagenic agents. And then he screened for worms that would like live much longer than they normally do. But he didn't know what genes were affected by these mutations. But he was the first to isolate longevity mutants in C. elegans. And then other people tried this in yeast cells and things like that. And so then later on, this like in the 90s, this really led to a bunch of high-profile discoveries. Like here's a paper in Nature where Cynthia Kenyon's team found a C. elegans mutant that lives twice as long as the worm normally does, which was huge. And then later on, people like Gary Rufkin and other people, they cloned these genes. They found the identity of these mutations. and they figured out in which genes these mutations had this effect. And they found genes in the insulin signaling pathway, for example, to have big effects on lifespan. And the next question was like, okay, this is a worm. I mean, who cares about C. elegans? What about more complex organisms? And so the next more complex organism they looked at were like fruit flies. You can still say, who cares about fruit flies? But the same mutations, I mean, in homologous genes in the fruit fly had the same effect. And so then people thought like, okay, that's really interesting. There's apparently genes that can help to determine how long we live. And mutations in the same genes in different organisms have similar effects. And later on, people also did this in mice and rats and so forth. So in humans, we can't do that, but we can look at centenarians, people who become 100 years old or older, and we can sequence their genomes, and we can identify factors that make these people more prone to become long-lived. And again, in many cases, this insulin signaling pathway is implicated. This guy here was actually my postdoc advisor when I was in the United States. And so this is a paper that made him quite famous. So it's like people started to discover these sort of pathways, which often have to do with metabolism, that harbor genes that, if mutated in a particular way, can make certain organisms more long-lived. And so here is this sort of insulin pathway that we also have in our bodies. We all have an insulin receptor at the cell membrane, and then there's this intracellular signaling cascade that leads to the activation of a transcription factor, which is called FOXO. And mutations in many of these genes, when you sort of reduce insulin signaling, tend to be beneficial in terms of how long organisms can live. And that seems to be conserved between worms, flies, rodents, and maybe even humans. In fact, in centenarians, people that live 100 years or older, many centenarians have particular mutations in this FOXO transcription factor. This is not the only pathway. People then started to realize that there's many pathways with peculiar genes that, if mutated or if overactivated, tend to affect how long we can live. And so then it became really possible to begin to study the genetic basis of lifespan. But so this is what the molecular biologists do, and their hope is, if we can tweak these pathways, maybe we can come up with therapeutical interventions that will make us live longer. So people, like there's like this book that was, I talked with someone about, with Gui before the class, by David Sinclair, who's a researcher at Harvard Medical School, and he looked at the drug that is called resveratrol. This is a compound, resveratrol, that's actually enriched in the grapes, in the grape skins of certain red wine grapes. And if you isolate this, it's like a polyphenol. It tends to have very good lifespan benefits for flies and worms, and then they did mouse studies. And so then Sinclair and other people tried to find similar compounds that are even more potent and to figure out their molecular mode of action. And nowadays, there's quite many drugs that people are hopeful about, that they might improve how long we can live. And so they're testing them in the fruit fly or in mouse models or even in primates. Another one is called rapamycin. But the problem is that these things might also have side effects and molecular consequences that we don't know about. And so it's not so clear whether we should like have this hope that we can develop these therapeuticals that will really help us to live much longer. So some people want to develop what is called a polypill, like combining different compounds that could make us live longer. But I'm a little bit skeptical about this. But so for the evolutionary biologists, the questions are a little bit different. Of course, the mechanisms are interesting and super cool and so forth. But for us, the question is more, what is the identity of naturally occurring alleles that affect variation in lifespan, for example? And if we can find such genetic variants, might they mediate trade-offs? Might there be costs of living longer? And to what pathways do they belong? Because this is like, let's say, sort of the molecular picture maybe of aging, and we can find mutations. But these mutations that people have studied on the previous slide, these were loss of function mutations or lab-induced mutations. These are not the sort of mutations that we find in natural populations, because these are mutations of big effect. But what about natural variation in lifespan in humans or flies or other organisms? Can we find more subtle allelic variants that might affect these things? And if we do, will they fall into these canonical pathways that have been worked out by molecular biologists? So that's like a big question. And so naively, one might think, okay, so these variants that contribute to the evolution of lifespan, they might fall into these classical pathways that were discovered by molecular biologists, right? But that's to some extent an open question. And so what are the effects of these alleles? And to sort of study these questions, me and other people got interested in using fruit flies, in some cases also other organisms, to let these organisms evolve a longer lifespan, and then to use population genetics and evolutionary biology to sort of understand the evolution of this trait at the genetic level. And so this is possible in short-lived organisms. We can't do this with humans or elephants, but with fruit flies we can do it. Because the average fruit fly lives something like 35, 45 days in the lab at 25 degrees Celsius. So they're short-lived enough that we can do experiments and experimental evolution. And this was actually done already in the 1980s. So this guy here, Michael Rose, who is a professor now at UC Irvine in California, and in parallel, another group led by Leo Luckinbill and Bob Arking, in parallel, these two teams tried to do a selection experiment in fruit flies where they tried to breed fruit flies to become more long-lived in a selection experiment. And they managed in both teams independently using slightly different experimental designs. And these are two back-to-back papers in the same issue of the journal Evolution. And this is the first demonstration that you can make lifespan or longevity evolve in the lab, in a breeding experiment. So as short as like 10 or 20 generations of selection are sufficient to produce flies that live genetically and phenotypically much longer than the baseline population. And so before I show you some data, I would like to thank the people who actually did this work. So this is a former PhD student of mine who is now a postdoc in Norway. And this is another former PhD student of mine who works now in the biomedical industry. Former postdoc of mine, Martin Capoon. And then Bob Arking collaborated with us. He's like the guy who participated in this study in 1983. And actually, Bob Arking kept these selection lines alive since 1983. So there's like hundreds and hundreds of generations of selection where these flies were selected to become longer lived. And we thought, like, rather than doing our own selection experiment, it's really tedious and hard to do and it takes time, maybe we can get his material and maybe we could look at the genomes of these flies. And so that's what we did together with these people, and then we also had help from Bruno Lemaitre and a bunch of other people. So what we did is like really simple. We thought like, okay, we're going to take these flies from Bob Arking. The first thing we did is we measured again whether they're still long-lived. So in that experiment, they had several control lines. So everything was founded from the same base stock of flies, which was then split in independent replicate populations. And so what Bob sent us were like 2 control lines that sort of have normal lifespan and 4 selection lines that are long-lived. They live something like 1.5 times longer than the control lines. And so in our hands, we measured these phenotypes again to make sure that this evolutionary differentiation persists. So we got 2 control lines, 4 selection lines. For the selection lines, this corresponds to something like 150 generations of selection. For the control lines that are shorter-lived, it's even more generations, something like 300. And the median lifespan for the females in the selection lines was 62 days, or 63 days, let's say. And for the males, it was 72 days on average. But this is almost twice as long as the controls live, right? So it's like, it's okay, let's say 40% lifespan and 50% lifespan increase in the females and the males respectively on average, which is huge. I mean, think about the human. Life expectancy for women in my country is something like 82 years now for women, life expectancy at birth. So think that, multiply that by a factor of 2, that's huge, right? So we took these flies and then we subjected them to Illumina sequencing. So essentially you extract DNA, which is easy to do, and you ship it off to the company, they put it in this machine and you get your sequence data back. So we did that by sequencing pools, you get 101 base pair reads, and we had a coverage of 230 to 260. And so then we tried to sort of figure out what are the genetic differences now between the control and selection lines. And we can do this in different ways, using methods that we've talked about yesterday, for example. And one thing that we can do is to calculate, for example, FST, which is this measure of how genetically different populations are, right? If FST is 1, it means that every individual for a particular nucleotide position has like 1 base, like the A, and all the other individuals in the other population might have a T. So that would be a FST of 1. And so in our data set, we said like, okay, we're on a fishing expedition. We don't want to be too stringent. We're not only going to look at fixed SNPs, but we want to look at SNPs that are very different, right? So we didn't look at FST 1, but like arbitrarily we said like, we're going to look for things where FST is 0.9, which is very, very strong genetic differentiation. Again, this does not prove selection, but this is an experiment. We have the advantage that we know what has evolved and who has evolved, right? We know that the control lines, they're probably subject to some selection, but mainly genetic drift. And we know that the selection lines, they must have evolved lifespan. And so we thought, okay, so what can we do here in terms of statistics? And we said we're gonna condition the data such that we're gonna compare each of the control lines to all the selection lines. And we're looking now not for idiosyncratic things that, for example, control line 1 might be very different from selection line 1 for some loci, but control line 1 might not be very different for some other loci, with selection line 4. But like we conditioned the data such that we did all these pairwise comparisons, conditioning the data such that like we look for consistent differences between the control and selection lines across these 8 comparisons, right? Comparing control line 1 to all 4 selection lines and doing the same thing for control line 2, and conditioning the data such that we are looking for FST outliers in the genome where FST is greater 0.9. And then on top of that, we said like, okay, this difference needs to be statistically significant using a Fisher's exact test. And so if you do this, this also means that the control lines are not going to be different from each other. They're going to have a FST that is close to 0. So we really want to identify the things that are different between the control and selection lines. So we did that, and this is called the Manhattan plot. So in fruit flies, there's like 4 chromosomes. The 4th chromosome is not plotted here because it's tiny. It has something like 20 genes. No one cares about it. So most people just like look at these chromosomal arms. This is the X chromosome. The left arm of the 2nd chromosome, the right arm of the 2nd chromosome, the left and the right arm of the 3rd chromosome. These are the major chromosomes in Drosophila. And so what we are plotting here across the genome is like this measure here, which is essentially the average FST between selection and control lines. So this is the average across all these pairwise comparisons, always comparing control to selection. This would be FST among, the average. And then we thought, okay, we're going to divide that by the FST within the control and selection lines. Because among the selection lines, if I would compare only the selection lines amongst each other, there might also be positions that are fixed or have very high genetic differentiation for reasons that might not have anything to do with selection. I mean, when you do these selection experiments, the population sizes are not huge. So to a certain extent, these flies will be inbred, which can also lead to high FST. And similarly, you can also get fixed differences or strong genetic differentiation among the control lines, and we wanted to sort of account for that. And so we sort of calculated the average FST within control and within selection lines. And so we came up with this measure, which is like a log 2. So 0, the 0 line would mean there's no difference in differentiation between control and selection lines relative to the differentiation within the lines. But if you go up here to this ratio being close to 1, this would correspond to extremely strong differences between control and selection lines, right? And you see a lot of red here across the genome. So the answer is, yes, there's genetic factors that might contribute to the evolution of lifespan, and they're distributed along the entire genome, right? So essentially we found like 8,000 candidate SNPs in 868 genes, which is a lot, right? So these genes might really be our best shot candidates for the evolution of lifespan. And so one question is like, what are they? But another question is, maybe there's many false positives in these data. How can we figure this out? And okay, so the other thing that we saw is like the candidates cluster non-randomly in specific genomic regions, and these things are distributed across the entire genome. But to be really sure that the data are solid, we did another statistical analysis. We wanted to ask whether these 8,000 candidate SNPs, whether they're different from random expectation. Now, the cool thing with this experiment, even though we had only 2 control lines and 4 selection lines, you can do a lot of informative pairwise comparisons, right, in this data set. And in principle, we can imagine a situation where we do all pairwise comparisons in the data and see what it gives us. And so this is what we did here. So to validate the SNPs and exclude randomness explanation, we used the combinatorial approach. So we can ask the question, how many sets of 8 pairwise comparisons are there if you try to compare 2, if you have this number of lines, right? Because like this is one particular set of 8 pairwise comparisons, in this case, comparing each of the control lines to each of the selection lines. But there's other sets of 8 pairwise comparisons. How many such sets are there? There are 6,435 possible combinations of 8 pairwise comparisons. And we're only looking at one that we deem to be biologically informative, comparing each control line to each selection line, right? But there's other ways of doing it. And so here, this is the number of candidate SNPs that we got in our analysis, which is roughly 8,000, using one set of 8 pairwise comparisons that's biologically informative. And here is the number of SNPs you get when you do all the other comparisons. Most of these comparisons are nonsensical in terms of biological information. And so you can sort of calculate the probability that you get this red bar by chance. The probability the set of candidates is due to chance. Okay, this is not a hugely small number, but it's 10 to the minus 4. I mean, so it's very unlikely, in other words, that you get this number by chance. And so that convinced us that this is probably an informative way of looking at selection. Now, the other thing that we did is we compared our data set to other studies. So we're like working 2 PhD generations on this project, which was maybe not that great for my first PhD student who started the work, because like it took 8 years to publish this paper, and we thought like, okay, we want to, because we analyzed the data, but then we thought like, we should not just like analyze the genomes, we should like do some experiments, and this is time consuming. And in between, in the meantime, we sort of got scooped by other teams doing something similar. They used other selection lines in Drosophila, not ones that we had access to, but they essentially analyzed their genomes to do the same thing. And we said like, ah, shit, right, that sucks. But we thought like, this is actually an opportunity. It's an opportunity because what these people cannot do is compare the data sets. But now we can take our data and we can ask the question, do we see overlap between the data sets? And we really wanted to do experiments, not just genomics. So there's like 2 papers that came out, one in Evolution, one in PLOS ONE, using 2 different selection lines for longevity, distinct from ours. And so we compared our candidate genes and our candidate SNPs to these studies. And what did we find? We found actually an overlap at the level of the candidate genes and even at candidate SNPs. So this is a small number. But you have to imagine this is a super complicated phenotype. It's probably even more complicated in body size. So there must be hundreds or thousands of genes in our genome that sort of contribute to determining how long we live. And we found 20 genes that were shared across these 3 studies. If you do a hypergeometric test of enrichment, this is actually significant at the 1 in 1,000 level, so 10 to the minus 3 p-value. At the candidate gene level. And yeah, so this suggests that like some longevity candidate genes exhibit genetic parallelism across studies. And these were different populations of flies that were used to do the experiments. The experiments were done differently in 3 different labs, but there is some core set of genes that was shared, which we thought is really interesting. And that made our paper much better than these other 2 papers, I would say. So then we did gene ontology analysis, which is an analysis. You get a list of genes, and now you might want to ask, what molecular functions do these genes have? So you can do a gene ontology analysis. And the interesting thing is, so we thought, when we did that, when we started this experiment, that what we're going to find are all these classical lifespan genes, insulin receptor, FOXO, AMPK, TOR. There's like a gene that's called INDI. INDI stands for I'm not dead yet. The first longevity gene that was discovered in Drosophila is called Methuselah. It's like a G-coupled protein receptor. Just like all these things that we knew from molecular biology. So when we started this, I thought like, okay, that's what we're going to find, right? We're going to find evolved alleles at these loci that are known to regulate lifespan. What did we find? We barely found nothing in insulin and TOR signaling. We barely found anything at all in these classical longevity pathways. Instead, our gene ontology analysis told us that what had evolved are, in particular, genes that have to do with immunity. We found an enrichment of genes that have to do with immune function. Now, it's of course sort of intuitive that a good immune system might help us to live longer. I mean, we can dream up things. But like immunity genes were not classically known to be involved in the regulation of lifespan. There were like a few papers where people asked about lifespan effect of immune genes. But not much is known about the connection between immunity and lifespan. But this is where we found statistical enrichment. So among our top candidates, we found approximately 30 immunity genes that showed extremely strong genetic differentiation between control and selection lines. Then we looked at the data from Remolina and Carnes, and then we saw that these guys, without really noticing it, also had found quite many immune genes. But they hadn't really seen it in their own data. And so many of them were shared across the 3 studies. And so here are the molecular immune pathways in Drosophila that also exist in mammals. These are not adaptive immune pathways like we have. We have an adaptive immune system. These are innate immune pathways, but we also have innate immunity. And so there's like 2 major pathways. Actually, the people who discovered the involvement of this pathway in immunity won the Nobel Prize. Actually, this guy here, Bruno Lemaitre in Lausanne in Switzerland, he discovered that this gene TOL, which is a receptor here, TL, at the membrane, that this TOL gene, which was discovered previously, that this thing is super important for immune function in Drosophila. So I think he was a PhD student, he had a Cell paper, like in one of these high-profile journals, Cell, where he looked at the function of TOL in innate immunity, and then later on, his supervisor's team discovered that TOL receptors in mammals also regulate immunity. And this guy, Hoffmann, he won the Nobel Prize for this discovery. But Bruno Lemaitre, who was the first author on the Cell paper, did not get the Nobel Prize. So he was quite pissed about that. But so, okay, there's this famous TOL pathway. And there's a parallel pathway in flies that is called the IMD pathway. The TOL pathway responds to infections with fungal pathogens or gram-positive bacteria, and the IMD pathway responds to gram-negative bacteria. And so you have this signaling cascade. And so here in red, along these two signaling cascades, are genes where we found many, many evolved SNPs in these genes. So not just like one gene in the toll pathway, but here things upstream of the receptor, the ligand, the toll ligand Spätzle, it's called, cactin, cactus, transcription factors like diff, and so forth. So we found like many, many evolved SNPs that were very different between control and selection lines in all these genes, and also some in the IMD pathway.
Okay, I find this very, very interesting. But the first thing is that I have a question, because we have an interplay between the toll signaling pathway and the AKT and mTOR pathway that was showed on the insulin via. And you guys have analyzed this, like this interplay between these two?
I mean, we haven't analyzed this in this study, which was not really a molecular study, but like a genomic analysis. But it's true that the insulin signaling pathway can regulate some of these things. Yeah. And so it's not impossible that you have a few changes in the insulin signaling pathway. Maybe you don't see it at the level of numerical enrichment of candidates. And so it could be that there's like a small change in insulin signaling that drives all expression changes or physiological changes in the immune pathway. But we have to remember here that even though such interactions exist, what we're talking about here, or what you have in mind, I think, are molecular physiological interactions. This gene in the insulin signaling pathway has an effect on the expression in the immune pathway. But what we're talking about here are evolved genetic differences, right? So there could still be genetic interactions or epistatic interactions between insulin signaling and this pathway. But the fact that we see all these distinct genetically evolved differences between control and selection lines in these pathways, I think, probably must mean something.
Yeah, and just to add two more things, is that I remember that in the beginning you were talking about the importance of studying the development as well to understand this, and the toll pathway plays a bigger role in the development.
Absolutely, that's how it was discovered, right?
Yeah.
That's how it led to the Nobel Prize for Janni Nüsslein-Volhard and Eric Wieschaus. And so in embryonic development, this pathway is super important, in fact. But in adult organisms, it is involved in immune function. Exactly. And so there might be a connection there, maybe. Yeah.
And I've never thought about it, but this pathway that's triggered by MYD88, it starts the expression of NF-kappa-B genes related.
Yes.
Are the ones
so these things here, relish,
they are
and they're like NF-kappa-B transcription factors.
And then like this is basically a strategy of the immune cells to become unsensitive to the medium and to work even with stress. Yeah. So it would make sense this type of pathway.
That this could be involved. Yeah.
In the
I just thought it's interesting. Like, I mean, if people do unbiased mutant screens in C. elegans or in flies for longevity, they didn't really fall on these genes. And so we thought like this is actually really cool, and the other papers didn't really highlight this. But still, is there, so this is getting to your question, is there now a causal link between the evolution of longevity and immunity? And I mentioned yesterday, I mean, or this week we talked a lot about these population genetic summary statistics that we can use to infer selection. And so now we could stop here and say, it's clear, these flies are long-lived, like what do we see in the genome? Statistically, we see this immunity genes. We can publish the paper, right? That's like, in a way, what the other people did in their papers, right? They just described the shopping list of candidate genes and then interpret these findings. But we thought, like, this is intriguing. We should, like, really see whether we can do something to show that there's a causal connection here, to do an experiment. And this is what I said yesterday. I think we can do population genetic theory and statistics all day long, but at the end of the day, we want to do an experiment. I mean, like the Higgs boson is beautiful. People knew that this might lead to a Nobel Prize. Peter Higgs might not have expected it, but it leads to a prediction. And the people at CERN and particle physicists, they wanted to see whether that thing exists. This is our business. It's like not to dream up theories, Our business is like to develop good theories, mathematical theories, and then test them against reality. And so we wanted to do an experiment. And so based on our genomic analysis, we came up with two completely dumb hypotheses, which is like selected and control flies have evolved differences in immune function, and these differences might have something to do with differences in lifespan. So how can we look at this? So the first thing that we did is we looked at gene expression changes or gene expression effects in some of the candidates and in some of the downstream effectors of these pathways. So both pathways actually, through these transcription factors, diff and relish, these bind to the DNA. What do they regulate? They regulate things that are called antimicrobial peptides, which are broadband antibiotics that we have in our body. So when we have an infection, our body produces these broadband antibiotics, these AMPs or antimicrobial peptides. So we wanted to look at some of these readouts of these pathways, and here we looked at three. This one is called drosomycin, this one is called atacin A, and this is called diptirsin. And so it's known from work in flies that as a function of age, in old flies, these antimicrobial peptides are expressed at a very high level, even when you don't infect the flies. And you think like, okay, that might make sense that the flies are old and frail, maybe they really need a good immune system and to express these things to not suffer from infections when these organisms are old. So that was very well known. But as a matter of fact, these old flies, even though they make more of these AMPs, they really suffer when they're infected. They have problems clearing out infections. They have high mortality upon infection, despite the fact they make more of these antimicrobial peptides. And so then people thought, okay, so maybe having a lot of AMPs at old age is actually maybe a manifestation of something going wrong with the immune system. Maybe the immune system is not very responsive anymore, and they try to maybe compensate by making many of these AMPs, or the AMPs, despite their high level, do not seem to be effective in these old flies. So this is known. And so this is what you sort of can see here. This is baseline expression of these AMPs. And you see here, we compare relatively young to relatively aged or old flies. And we see here in the control flies that don't live long, they tend to upregulate with age these AMPs, right? But in the selection lines that are long-lived, we didn't see that. The sort of reaction norm or the curve stayed sort of flat, or at least the increase was less steep in these selected flies as compared to the control lines. So in other words, there was like a significant age, young versus old, by selection regime statistical interaction, which told us that the selected long-lived flies have somehow a different age-specific regulation of these antimicrobial peptides as compared to the control lines. So the baseline AMP levels go up with age in the control lines, but remain roughly constant in the long-lived flies. So that already told us that there is really something functional about the immune genes that has evolved, and there is a difference in a way in terms of these immune readouts of these genetic changes. It's not just like, we see genetic changes that don't do anything functionally. So this was like a first indication. But then we also infected the flies, because this is baseline expression. So what about induction, the inducibility of these very same AMPs when you infect the flies? And so this is what we looked at here. And so this is sort of quantified here as log2 induction, the ratio of selected to control flies. And we saw that, the control flies, they tend to sort of upregulate the expression of these immune genes when infected as a function of age. When they're young, the selected flies tend to make more of these things when infected. But when they're old, they tend to downregulate these AMPs. In contrast to the control flies tend to upregulate the AMPs, but the selected flies tend to downregulate the AMPs. And so that makes you think that there's a big topic in human immunity, that the immune system, of course, is good. But if the immune system is too active, it's really bad. And there's this phenomenon that people call immunosenescence, which is the fact that the immune system is aging, but the immune system might also contribute to your own aging, especially if it's not appropriately regulated at old age, right? And so we thought like this is super interesting, that like the long-lived flies tend to do the opposite of the control flies in terms of regulating these AMPs. So then, okay, that's gene expression, but what about realized immunity, as we call it? Realized immunity is like when you infect the organism and you see how well it deals with the infection. And so here we infected the flies and we measured how well they survive, right, upon infection. And so we did this with young and old flies. The solid lines are the aged flies, control and selected, and the dashed lines are the young flies, again, control and selected. And what we saw, infecting the flies with a fungus, with gram-negative and gram-positive bacteria, and even with a specific virus, we found that pretty much independent of age for most pathogens, the selected flies are much better at surviving the infections when they're young and when they're old. And despite the fact that they downregulate AMPs when they're old, they're still better than the control flies at dealing with infections, which was very interesting. So the long-lived flies survive infections better. And again, this means that the genomic things we had seen in the sequencing data are not just false positives. Then we also looked at clearance. So one measure of immunity is like how well you can clear out the bacterial infection. You can measure the bacterial titers. You can do this using a bunch of things. You can, for example, grind up the flies and then sort of like, take aliquots and plate the stuff on an agar plate and look at bacterial growth and so forth. So here we looked at the proportion of clearance, and what we saw is like the thing that has been observed many times in regular normal flies, that flies that are young have a quite high ability of clearing an infection. So this is complete clearance. You could also look at partial clearance. This means 100% clearance, which is sort of an extreme measure. But so these guys can do this fairly okay-ish when they're young, but less well when they're old. But the selected lines, they had a significantly reduced clearance ability when young, and it stayed, statistically speaking, constant as a function of age. This led us to sort of hypothesize that maybe what these flies had evolved is not increased immune resistance, but maybe increased immune tolerance, right? And so the long-lived flies might have evolved improved tolerance. Even though this is reduced a little bit here, I mean, they sort of like maintain this constant clearing ability as a function of age, whereas the control lines, they significantly decrease their clearing ability. And so then finally, we wanted to see, okay, do these immune genes that we find here, can they affect lifespan? Now, nowadays, you can do CRISPR-Cas9, right? So with CRISPR-Cas9 technology, you can change single nucleotides. And I can ask for a particular nucleotide position. Does it matter whether I have an A or a T? And so evolutionary biologists are, including people in my lab, we're trying to use this technology to study evolved alleles. So we can use sequencing like this in population genomics, and then we can use CRISPR-Cas9 to test evolved SNP changes. We didn't do that here because this is actually technically quite challenging, and not many people are doing this in evolutionary genetics. I mean, molecular genetics, everyone is using CRISPR-Cas9, but they're not studying single nucleotide changes. They use CRISPR-Cas9 to knock out a gene or do other stuff. But like manipulating a single SNP and just that SNP in a fly where the genomic background is completely the same otherwise is not so trivial technically. So we didn't do that here. We did something that is less ideal. We didn't test the evolved genetic changes, but we just like said, okay, let's knock out these genes and see whether they have an effect on lifespan. This does not prove that the evolved changes, genetic changes affect lifespan, but it would show whether or not these genes, these immune genes, affect the traits that we're interested in. This is not as good, maybe, in terms of inference, but it's the next best thing. And so this is what we did next. And so, yeah, actually at this time, pretty much nothing was known, or not much, about how immunity affects longevity. So we knocked out or manipulated these genes here. Spätzle, which is the ligand of the toll receptor, cactus, and this transcription factor diff. And so how did we do that? We used RNAi, RNA interference, and you can essentially order fly stocks. So the fly genome has 14,000 genes. For all these genes, we can order mutants, or we can order RNAi lines. So we ordered these RNAi lines, transgenic RNAi lines, and using transgenic tricks, we can sort of induce RNAi in the fly, for example, when they're adult. Because you had mentioned that correctly that toll is a developmental gene. And we didn't want to use a toll mutant because, I mean, most toll mutants are lethal, embryonic lethal, right? Because their development is screwed up. You could take milder mutants that survive into adulthood and then ask, do these flies live longer? But then you might have developmental carryover effects from what the mutation did when the flies were developing. But so using fly genetics, we can play a trick. So we can knock out this gene specifically only in adult flies by feeding the flies a drug in the food that will turn on the RNAi, which will then silence the gene. This is way better than a classical mutant. So we did that here. And I mean, as you can appreciate here, the effects were really small and mild. But so this was a sort of adult-specific RNAi for Spätzle, the ligand, the receptor, and the downstream transcription factor. And indeed, we saw mild, not very massive lifespan extension. And so we can wonder, why is this not you know, a stronger effect? It's clear that in our experiment, many genes in the genome contributed to the difference in lifespan, and where we can't say like the immunity genes are the only ones. Maybe they sort of interact with each other synergistically. So these lifespan effects were rather mild, but nonetheless they sort of confirmed the hypothesis that these immunity genes might have to do something with longevity. But maybe more interestingly, when we look at cactus, what is cactus? Cactus is this thing here. And cactus is actually, all these components here are sort of positive regulators of immunity. But there's also in all these pathways inhibitors or antagonists. They're like sort of like a brake on the immune system. When your immune system is active, they keep your immune system in check by down-regulating a little bit. Or if it's really too highly expressed, they're like really sort of stopping it. So here, cactus is an inhibitor, it's an antagonist. It should have the opposite effect, right? And so we knocked out the inhibitor with RNAi. What happens when we knock out an antagonist of the pathway? The pathway becomes hyperactive, right? It becomes inappropriately active. The immune system is overshooting. And so this is here, when we knock out the antagonist, what happens? The flies live 60% shorter when you take out the antagonist. This really shows that like the hyperactivity of the immune system is really bad for how long you live. And so maybe this connects back to what we had seen here, the fact that the long-lived flies tend to perhaps downregulate some of these immune factors when they're old. So maybe what these flies have done is to evolve lifespan by sort of evolving another regulation of their immune system that allows them to be better at surviving infections when they're old by fine-tuning in a way the expression of the immune system, avoiding hyperactivation of the immune system when they're old, which often happens in old flies, and maybe evolving a better tuning, age-dependent tuning of their immune system. So this is what we had seen here, this massive reduction. And then, okay, there's a bunch of other papers mainly about the other pathway, not the toll pathway, but the IMD pathway, that had found sort of similar effects, not in the context of the evolution of longevity, but so these studies together with ours clearly show that there's some link between immunity and longevity, which is not that far-fetched, obviously, but this had not really been seen before in an evolutionary context. And I just wanted to show you this short story to sort of illustrate that, yeah, so population genetics and genomics is cool and we can do statistics and all these things. But at the end of the day, I think we should be doing experiments to test particular candidates. And we do other studies in my lab where we do similar things. So our business is really to do the population genomics, but then to do functional experiments to really verify that the candidates do something. And in some studies, we were using CRISPR-Cas9 to do that, or other tools, to really look at evolved SNP differences, or in another story that I won't have time to tell you about, we're doing this in the context of a chromosomal inversion, which spans 1,200 genes. So we can isolate this inversion and measure phenotypic effects and so forth to really demonstrate causally that selection is probably involved. So this is, yeah, sort of the summary. Yes, please.
This CACT inhibitor role, it's on the diff pathway?
It's in the toll pathway.
Yeah, but then can you just go back? Because, yeah, this thing, so, I don't know, for me, like, if you do the knockdown directly of diff, and you do not see a really great difference between the lifespan, and then if you do this in a upstream a kinase, upstream protein, and then you see it, probably this protein is acting in the other pathway, you know, like the impact of this, because
in this pathway? No.
No, like in the one of the left here, because if he CACT acting direct is a transcription factor down there.
Yes, yes.
Because I don't know, think that if CACT is inhibiting diff, and then you make diff knockdown, and diff is important to the lifespan, then you would see this difference.
Yes, I know what you mean. Like the effect on diff for diff was like much smaller than for cactus.
Exactly.
Right?
So
on the other hand, like I mean, what it can also mean, I agree, but what it also means is like this is a super strong antagonist, right? And like I mean, so we know from other experiments that we haven't done that when you inhibit the antagonists, you can measure that the immune system really goes through the roof, right? Which is really bad, right? I mean, so we sort of do know that, but I agree with you that there could be other things going on here. And I agree that like normally we would expect to see symmetrical effects in opposite directions in terms of the effect size for cactus and yeah. So of course we were like a little bit disappointed that these effects were rather mild, but I think they sort of went in the right direction. And so now it will be interesting to sort of figure out, so how do these genes affect lifespan molecularly?
In this hypothesis, the aging process will be basically linked to an inflammation-induced senescence.
Exactly.
Right.
So that's also what people think now about these AMPs being highly expressed in old individuals, that this is not good, right? But it leads to a state of, chronic inflammation, actually, because like the flies can't clear out the infection despite making more of this immune stuff, and they're actually sick. And somehow they can't really properly downregulate that thing. I mean, if that would be possible, they should do it, right? I mean, it's not good for their physiology. So somehow it's mis- or dysregulation. And I believe, I can't really prove at this stage, but I really believe that these long-lived flies have found a better way of fine-tuning this. And so what we could do in principle with fly genetics, but we haven't done that, with this system that we have here, we could knock down this is like knockdown through the entire adult lifespan. But we could do it at particular ages. We could turn it, do RNAi only in young flies and see the effect on lifespan, or we could like do it at a particular age and then ask how it affects the remaining lifespan of the flies. So we could like do age-specific manipulations, which we haven't done here. Yeah. So this is like sort of brute force, and we're turning down the immune gene during the entire lifespan of this fly. I mean, like, that might have detrimental effects as well, and that might obscure bigger differences or effects on lifespan. So it's not trivial to do these things and to figure out how they work. But I would say, like, in these experiments overall, we had pretty solid evidence that there's a link. So we found that long-lived flies have reduced induction of these AMPs at older age, but survive infections much better than the controls. Then we found that the long-lived flies might have evolved higher tolerance. We saw that downregulation of the immunity genes mildly extends lifespan, perhaps by reducing the costs of immune deployment or the costs of hyperactivation of the immune system, and conversely, chronic immune hyperactivation by knockdown of the antagonist really shortened lifespan quite dramatically. And so we concluded that longer lifespan can evolve, at least in part in this study, via evolutionary changes in immunity. And again, what was really interesting is that we didn't really see many changes in canonical lifespan genes, whatever that means. And so this is sort of what we did. So these are some of the, well, mainly former lab members in my team, some of which contributed to this. And yeah, so this is like sort of the end of the talk with this little cartoon where the nurse tells the guy, like, this is amazing. Researchers at Harvard found long-lived fly mutants. The guy couldn't give a crap. How does this help him, right? I mean, yeah. But it might help, actually. So that's what I wanted to tell you. I'm not going to start another lecture because I wouldn't be able to finish it. It's Friday. It was a long night for me. But it was a great week with you guys. I really enjoyed meeting you all. Thank you for all the questions and discussions. And yeah, it was a real pleasure being here and teaching this class. And I hope you will have continued fun till December. And then you will all go home to your families and celebrate Christmas. So it was super nice to meet you. Thank you very much.
I really want to thank you very much. It has been amazing. I'm pretty sure they enjoyed a lot. And yeah.
Thank you very much.
Thank you very much. So guys, unfortunately, I'll have to give bad news for you. I'm just passing the bad news, okay? It's not. So you don't have coffee break Monday. I'm so sorry for that. I tried to extend it, but I couldn't. So, yeah. But please, for the other forms, few of them, we want coffee breaks. Yeah, we are going to take the picture now. I'll call Natalia.